right, we're in business. Take it away, Jinghui. Thank you.
Okay, thank you so much, Mark, for the great introduction. And I'm really excited to hear that CICERO has made so much great progress in advancing precision medicine for pediatric cancer. I visited Australia about 3 or 4 years ago when CICERO just launching, and to see how much the impact the project is on pediatric cancer patients in Australia. We feel we really are working together globally to help cure the children and saving lives. Really excited to be here, joining every one of you. So today I'm going to talk about the utility of whole genome sequencing in clinical profiling of pediatric cancer. And I have no conflict of interest. So my topic is going to intertwine a little bit about both the genomic analysis in research and in clinical. So we all know that in normal process for developing clinical application is starting from research, studying a cohort where we can find prognostic markers predicting drug resistance and adverse effect, and test them on the cohort. But as we have moved more with clinical sequence testing as an integral component of oncology, many of the data sets generated from clinical could be reutilized for research purposes, and I'll illustrate this process during my talk. So first, I want to introduce a research project actually had a pretty fundamental impact on the platform we decided to use for pediatric cancer clinical profiling. This is a study that we completed in 2018 in collaboration with National Cancer Institute, the Target Initiative. From that study, we have performed a whole genome, whole exome, and RNA-seq, and performed extensive data curation and identified 142 driver genes involving pediatric leukemia and solid tumor. When we compared this data set with the 3 pen adult cancer studies at the time, we recognized that more than 50% of the driver genes are absent in these adult cancer patients. So this is indicating that pediatric cancer, we cannot use panels developed initially from the pan-cancer analysis of adult cancer to proceed in clinical. The second aspect is 62% of the variants we discovered are structural copy number variations. So that shows the importance of whole genome sequencing in the clinic. And I will elaborate a little bit more on some aspect of finding structural and copy number changes. So as we all know that clinical genomics is a multidisciplinary team effort involving both computational scientists as well as pathology, oncology, cancer predisposition, and disease expert. So we started the journey in 2013 with a pilot study to develop the pipelines and experimental protocols to do what we call the 3-platform clinical sequencing of whole genome, whole exome, and RNA-seq. So 2015 is we entered the phase of feasibility analysis in real clinic through the Genome for Kids protocol. 2017, we have added 2 additional tests. One is called Rapid Turnaround Time RNA-seq. This is to support the Total 17 leukemia clinical trial that uses kinase fusion status to determine eligibility of a kinase inhibition clinical trial in kids with leukemia. The second is FFPE-based 2-platform exome and RNA-seq. And 2017 is also we entered the clinical service. Right now, it is almost more than 5 years, and we have analyzed more than 2,000 cases utilizing this platform. So I want to give a brief introduction on the clinical pipeline that we used. This is developed through analyzing both the tumor and normal samples. And at the time, we have set the whole genome sequencing to 30x coverage. 30x coverage is the one that usually used for a research project. And we analyzed both the somatic and germline genomes for a variety of the variants that will be of interest. And then we perform variant classification utilizing a database that we have curated, including both the publicly available databases like Cosmic ClinVar literature, as well as a database assembled through our research project called the Pediatric Cancer Genome Project, PCGP. From there, the analysts will work with the panels to determine pathogenicity of these variants, and pathogenic and likely pathogenic variants will be signed out as tumor report to the physician and germline report that will be communicated by the genetic counselor to patients and families. So I want to give a little bit more detail on the pediatric cancer, the variant classification pipeline we created called PCOMPI. PCOMPI is also very well known as a famous dish in Memphis. This is where my institution, St. Jude Children's Research Hospital, is located. So what it does is that can take the variants either detected from whole genome or whole exome analysis and whittle it down to through different processes, including candidate gene filtering, annotation, population filtering, and automated classification using the database that we created. And it classified these variants by assigning metals called gold, silver, browns. And as this tool was developed during one of the Olympic Games, that's why we have these medal assignment for the variants. And the variant then can be, medal variant then can be analyzed by the panel for further classification. And the system was initially developed for our 2015 New England paper for germline cancer disposition in pediatric cancer patients. We did a follow-up study published in JCO on pediatric cancer survivors, and was also used for our feasibility study, Genome for Kids, which I will discuss a little bit more. And the tool has also been utilized for research purposes. I just want to mention one publication in collaboration with Dr. Jun Yang from Pharmaceutical Science. And he was studying familial leukemias, and using the standard linkage analysis, he was not able to detect a candidate gene. But PCOMPI actually going through the exome data only nominated one gold variant, which is a loss function mutation in ETV6. And we later recognized the reason that this variant was not picked up by linkage analysis is because the patient, one of the kids, have incomplete penetrance, so the phenotype analysis missed this one. When we were preparing PCOMPI, Theo Susi from Sweden mentioned that we should really incorporate functional assay data that has been available for p53 mutations in both mouse models and yeast systems. So we have designed an interface that incorporates the data he has collected from literature into the system, and we anticipate similar functional data generated from other studies can also be incorporated as part of the variant classification system. And this whole pipeline is not only eligible for, applicable for pediatric cancer patients. We have recently collaborated with Marcin Wildawski from Hematology Department, who focuses on studying children with bone marrow transplantation failure. And we were able to modify our candidate gene list to include the BMF candidate genes and compare these with clinical testing results. And we were able to replicate all the findings that people usually have to sign out with multiple panel testing to get a clinical diagnosis. Gene fusions, we used a pipeline we call the CICERO for detecting fusions. And for clinical use, we also implemented a cloud-based version to ensure that for samples with a high number of structural rearrangements like osteosarcoma in pediatric cancer, that we can predict gene fusions at a high speed by utilizing the CPUs on the cloud to speed up the analytical process. And this is some of the benchmark data that shows that CICERO is very good at not only finding the canonical highly expressed chimeric transcripts, but also can identify variants like involving non-canonical fusions, such as enhancer hijacking and internal tendon duplication ITD events, which are very important for pediatric AML. So in terms of looking at the diagnostic yields, we have compared our 3-platform analysis with single platform and dual platform exome and RNA-seq. And we found the power of whole genome where it really gained is in detecting focal copy number changes that encompasses low number of exons or have very small exons. And these are the areas where exon-based capture basically lack the probes to give robust call on these focal copy number changes. And these are the 2 examples that we've seen that were missed by exome-based analysis. One is a 22 kb germline deletion in a medulloblastoma of Sarcomas hedgehog subgroup, as shown here. And the other is a DMT3A deletion in a neuroblastoma that involving deletion of exons 3 and 6. So what we learned from this pilot study is that we did see about 2 cases that we missed the variant. One is a BRAF mutation, and the other is a BRAF KIAA fusions in low-grade glioma. Some of the reason is that the gene expression level is very low, fusion expression level is very low, or the mutation is highly subclonal. So when we then performed a simulation analysis to look at if we increase the whole genome sequencing to 45x coverage, what we can gain from the variant detection. What it shows here is that at 30x coverage, we are able to have 90% of the power for detecting variants with BAF of 0.1. And if we increase to 45x coverage, our kind of power can be increased to 99%. So this is when we moved to the Genome for Kids protocol. We decided to move the coverage to 45x whole genome. And I also want to share an interesting anecdotal, like when we were trying to get ready to publish Genome for Kids, we're coordinating with Mark and Paul, trying to see we can do back-to-back. And I have to say, Zero beat Genome for Kids in that forum, in that Nature Medicine decided to take Zero. And we ended up having to resort to cancer discovery and was published last year. So we are a little bit behind in publication schedule, but it was really exciting to see that, the message is consistent, that we are both institutions felt like moving towards a genome-based profiling is really going to make impact on pediatric cancer. So I want to share a little bit about the infrastructure, how we do the G4K clinical testing. So the patient samples were obtained, and a portion of these still goes through standard molecular diagnostic testing, including histopathology, like histology, IHC, as well as ancillary tests, FISH cytogenetics, SNP-array, RT-PCR, Sanger, flow cytometry. Some other portions of the samples, fresh frozen raw, go to clinical sequencing. And we also have to checking the patient consent to make sure that they agree to perform clinical sequencing for both germline and tumor samples. So we will perform the genome-wide profiling. And for CNS tumors, there's also a methylation array performed for classifying the CNS tumors. And then when the data were generated, they actually will be moved to a research team doing the clinical analysis. So at this time, we move remove PHI, MRN, and all the analysis was run in an isolated clinical environment locally. And the genomic data were analyzed as I described previously, and results will be discussed and correlated with other ancillary tests together and reviewed and signed out back to the EMR. So for Genome for Kids, there's about 306 cases total, 300 for germline testing and 253 for tumor testing. And we have captured major subgroups of pediatric cancer as shown in the pie chart here. So in this gray bar, which marked as others, there's actually 18 different rare subtypes that has less than 200 cases annually per year in the US that was used for, that was captured through the Genome for Kids program. So what's the effect of increasing the coverage to 45x? And this is a variant allele fraction comparison between the whole genome and whole exon for SNV and for indels. And we could see these hotspots here are pointing to the low frequency, low VAF areas. So we actually have 41% of variant have a VAF below 0.2 and 15% have a VAF below 0.1, really indicating that increased coverage really helps with the sign out of detecting of these subclonal variants or handling tumor samples with low purity. And these are the somatic findings that we sign out, and for in leukemia, solid tumor, and CNS tumor. So we have color-coded variants by copy number, ITD, SME indels, SV, and fusion genes. And we could see that colors representing structural variants here actually account for the majority of the clinical sign out. Again, showing the importance of genome-wide profiling. So gene fusions are probably the class of the most valuable class of the biomarkers in pediatric cancer in terms of subgroup classification, as well as clinical actionability. And we have indeed find the common fusions in, as shown here, in different diseases, most notably in leukemia samples. We also find the rare fusions that in, in all three different cancer subtypes. And I want to point it out, you can see the leukemia cases actually diminished. A lot of novel fusions were detected in rare pediatric solid tumors. And we also have utilized the whole genome sequencing data to find the enhancer hijacking event in leukemia and brain tumor. So I want to showcase two examples here. The first case is a child with metastatic melanoma who had failed multiple therapies, including immunotherapy. And the patient does not have BRAF mutation or BRAF fusions. So when we received the tumor, it was subject to 3-platform sequencing, and we have discovered an in-frame MAP3K GNG2 fusion. And because the last exon of MAP3K encode a autoinhibitory domain, so we predicted this to be an activating mutation that will activate MAP kinase signaling independent of BRAF. So MAP inhibitor was recommended. The patient had total response, but later developed resistance and passed away. So we were actually very fortunate, also was able to receive the relapsed cases, and we analyzed. No additional driver mutation was discovered except for one copy gain of the fusion event. So upon seeing this event, we collaborate with Amita Baharamani from pathology, we have a collection of FFP spheroid melanoma. So we performed a RNA-seq on these collection of the samples. And to our surprise, MAP3K8 gene fusion or truncation actually was the most prevalent variants discovered in this cohort, and it is mutually exclusive with other type of the variants like kinase variants, mostly gene fusion event detected in this cohort. And interestingly, they all affect exon 9, and we all predicted this all will lead to the loss of exon 9 in MAP3K8. We then looked at examining the RNA-seq profile and find indeed exon 9 expression is much diminished compared with exon 8. So using this one, the expression of the exon as a signature, we were very quickly able to go through the TCGA melanoma data that were hosted on the GDC portal. We actually could just use the band slicer to cut down this gene and looking for differential exon 8 and exon 9 expression, and was able quickly to identify about 8 cases that have reduced exon 8 expression, and later on we're able to run a full RNA-seq gene fusion analysis on these cases, and they all identified the fusion event similar to what we found in adult cancer. So this fusion is present in 2% of the adult melanoma, and there has been increased interest in developing inhibitors or applying inhibitors that specifically targeting MAP3K8 in pediatric melanoma. So the second example is in a T lineage AL that involving transcription factor TLX3. The canonical fusion activation mechanism is caused by gene fusion between BCL11B and TLX3. So in this particular case, we're actually seeing a fusion involving chromosome 5, which is where TLX3 is, and chromosome 8. And the chromosome 8 is the intragenic region encoding a known enhancer of CIMIC that's called NME enhancer, as shown here. So we were able to find by doing, this is the event that discovered utilizing a new software for non-coding variant analysis called cis-X, which I will introduce a little bit later. And when we perform gene expression clustering using TisNeMap, using the TAL data that we previously analyzed in the research cohort of NCI target, we do see indeed a cluster with cases that known to have the TLX fusion. So the cis-X was published 2 years ago, and the study was led by my former postdoc, Yu Liu, who's currently at Shanghai Children's Medical Center and had his own laboratory. The idea is that if we want to find cis-activating mutations, one of the signature we do is we can use polymorphic variants or heterozygous SNPs or somatic mutations that in the DNA, we can see heterozygous alleles. In RNA, we see monoallelic expression. So in order to develop a statistical model, we have to establish a null hypothesis to compare with the pattern we observed is consistent, it deviates significantly from the null model. And the statistical model we use is using a combination of Gaussian plus binomial, and we can show that in both of the RNA-seq sites with low coverage as well as those with high coverage, our model outperforms the simple binomial model or beta binomial model compared with the truth, which is experimental data. And for this analysis, we focused on outlier high activating mutations. And this is because outlier low can have multiple interpretations, such as imprinting methylation or nonsense mediate decay. So we start with something more straightforward, which is a pattern of activation. And this is how pipeline looks like. And I just want to mention that we also utilize the reference expression matrix, collecting from research cohort to determine the outlier expression, and use the TAD domains as well as any other functional data to determine the somatic variants could be considered as the cis-activating non-coding variant. And the benchmark analysis we performed was using T-lineage AL. This is because there has been rich knowledge collected from individual researchers, discovering one location at a time through various intense functional-based approach. So we have sufficient variant to compare with a positive control to compare with to benchmark this. So we found about 90% of the benchmarked genes in a cohort through a collaboration with Shanghai Children's Medical Center. And we were able to also discover novel non-coding variants and verify them from both the in vitro assay as well as in vivo PDX model by performing ChIP-seq analysis. Now I want to switch gear, talk about some of the interesting germline variants that we reported in the Genome for Kids cohort. The first is a MutYH G396D mutation, and this is a founder mutation known to be contributing in colorectal cancer. But in this particular case, we actually found this in a variant in a retinoblastoma patient. Interestingly, there's also a second LH event leading to loss of the wild type allele for this gene in the tumor sample. We noted that this sample, the retinoblastoma sample, is unique in G4K in that has the highest mutation burden across all the retinoblastoma cases we performed in Genome for Kids. And we also, when we performed the mutational signature analysis, we noticed that it has a ROS signature, which is consistent with the loss of repair functions involving MutYH in this pathway. So what we believe that under the specific circumstances that this MutYH variant is relevant for the disease. The second is a non-hotspot mutation, A161T, in the germline of TP53 in a ganglioneuroblastoma patient. So over here, we are using the red and the blue bars indicating reference versus alternative alleles in the germline DNA over here and in tumor DNA. So we do not see loss of heterozygosity in tumor DNA. But in tumor RNA, we only observed the expression of monoallelic expression of the mutant allele. And using the AFC test, a statistic we developed for cis-X, the non-coding variant one, that we were able to show this is a highly significant pattern. And histology, the subsequent IHC analysis confirmed the observation we have in the RNA-seq. So we nominated this, given the biallelic loss, perhaps due to epigenetic silencing of the wild type, we nominated this as a pathogenic variant. Finally, people ask about what happens to these patients, because in the G4K kids, we performed the profiling on every child treated at St Jude with fresh frozen material. And so 85% of these patients were already enrolled in clinical trials. So we really cannot change the therapy on that. So the delayed publication actually give us some benefit that there are subset more patients relapsed. So we can look a little bit more on the treatment changes that happen in these patients. So these are the swimmer plot where about the 15 cases where there's a genomically matched therapy changes that happen to these relapsed patients. And we can see that most of them still succumbs to disease. Only 2 survived. And I'm not discouraged by this result. As in the case of the melanoma MAP3K8 fusion, is we feel like this really argues for that genome matched clinical testing result really need to be incorporated as the diagnosis, not only used for relapsed and recurrent disease where patients has already been in a poor condition to receive the therapy. So some of the kind of trials specifically designed for newly diagnosed patients like Total 17 will provide more accurate, informative results than only those cases, only those results that we obtain from relapsed and recurrent disease. So a question we were always asked is how good the 3 platform is versus the gene panel. So this is the end we performed a analysis only based on the gene identities, compare our result with Foundation CDX, Foundation Theme, OncoMine, Heme, OncoMine, OncoKids, and looking at both the P and LP variants, actionable variants. And also we look at the patient level, how many of patients will miss at least one reportable variants. So Foundation Heme is the best platform. We only look at the leukemia. They captured 84% of the reportable genes. But when we look at individual level, there's still about 1/3 of the patients that have missed a reportable variant. So again, I just want to mention that Genome for Kids is a massive effort with many contributors across different departments. Dr. Kim Nichols is an oncologist who wrote the G4K protocol, and her team also performed germline cancer susceptibility analysis and communication to patients and family for this study. So our future direction, including improving the turnaround time for 3 platform sequencing, incorporate RNA-seq based classifier as an integral process of tumor subtype classification, and improving sensitivity for detecting early expressed gene fusion, expand germline structural variation analysis. And I want to give a little bit more progress report on the RNA-seq based classifier. So this is a process we have created, which we actually will utilize RNA-seq data on the research environment, perform multiple curations, including ontology classification, harmonization of data generated from different platforms, different protocols, batch correction, and then generate reference maps by t-SNE plot. With these reference maps for RNA-seq that has questions on the clinic, we can actually compare the expression profile with those on the t-SNE and find where they group closest together to gain insight on what are the, resolving some ambiguities with the cancer subtyping. And this is work led by Alex Scout, who's an Australian and joined my lab about 3 or 4 years ago. And he's the one who is really leading many of the RNA-seq based activity. And the reference map, I put a link here, is accessible through our St Jude data sharing cloud. And this is the reference map that we created, Alex created for the pediatric solid tumor that we have profiled in the research setting. So one of example is to show the expression map is able to, in the real clinical use, to resolve fusions in a NOS of round cell sarcoma. And this is a sample that we only have RNA-seq on the FAPE and reviewed a fusion called with a very unrecognizable name, but both of these fusion partners are pretty close to a known fusion. This is located pretty close to DUX4, and this gene is very close to SIG. So when we run this, TSNE analysis pipeline, we can see this clinical patient sample very closely located to its closest neighbor. Two samples we profiled with a fresh frozen material and show SIG DUX4 fusion. And then the clinical lab is able to use SIG break apart fish to confirm rearrangement of SIG. And this is also shown a very high expression of SIG in this one. So this case was signed out with confirmed SIG DUX4 fusion in that case. I just want to use the last few minutes to talk about the data sharing and visualizations on St Jude Cloud. And this is the platform we developed to support research in pediatric cancer, pediatric cancer survivorship, as well as non-cancerous diseases like pediatric sickle cell diseases. So I want to mention that all G4K samples, as well as over 2,000 real-time clinical samples, are now available for research. And even though pediatric PCGP, our research study was started earliest in 2010, we are seeing now the most requested data sets by the users are in all the clinically generated data set, and really highlighting how these being used not only by St. Jude researchers, but by people across the globe. So we decided to put the data on the cloud-based platform is because that will ensure that people who do not have the resources for data storage or computational infrastructure can port their favorite analysis, either utilize the analytical tools on the cloud already, or perform the analysis, or port their tools on the cloud and perform the analysis. And there's extensive curations that we have performed, including data harmonization, variant curation, metadata collection, and expression matrix. So we have generated 3 apps. One is called PCON, the pH Cancer Knowledge Base. That include all the published data, not only generated by St. Jude, but by other institutions, that we hope that the zero data can also be part of this at some point. Genome for Kids genomic platform. This was built in support of, supported by Microsoft, Azure, and DNA Nexus. Microsoft has provided a generous support for providing the storage for this platform, and visualization community with all the visualization graph, and the TSNE plot I showed earlier was part of that. And this is a stat, 1.2 petabytes of data, 400 requests from 125 institutions. And we also have about 3,000 registered users for cloud-based analysis and 2,500 unique users per week for public data access. So again, given that we profile every child at St. Jude, there's a lot of the rare subtypes of PH cancer that was captured through clinical genomics platform. And the process I showed earlier about TSNE map generation is the same that we used to transition from research to clinical setting. So what people can use this for, I want to mention one use case. This is a patient, Tumors. There's a company was interested in determine clinical trial eligibility for homologous recombination deficiency, and want to see how many patients in our cohort have that profile. So we were able to use the SME indels and copy number data from the whole genome. This is important because the overall mutation burden in PH cancer is low. So exome data, you won't have the power to analyze robust signature. And then we performed the SCAR HD analysis using CNS data and combine these together to get a HRD score. And also overlaying these HRD genes, either disrupted as part of our germline cancer predisposition gene or somatic rearrangements. And we're sending these prevalence data to FDA as a collaboration with the company. So this is my last slide, and I want to say this is a team effort that most of the people worked at home during COVID, but it doesn't slow down our progress. And I want to mention that Clay McLeod here, along with Alex Scout in the background, are really instrumental to ensure that all the data has been made publicly available, and the Genome for Kids and the ClinGen data has been accessible for research use on St Jude Cloud. Thank you very much, and I'm ready to answer questions if there's any.
Jinghui, that was just spectacular, and thank you so much for joining us. I really wondered, what's your review process? I think one of the things we're finding is that so much of these genomes are complex and there are so many features we don't understand. So how do you go back and re-review all the data to find new things or things that are coming up, or variants of unknown significance that now shift to being more significant or less significant? Do you have a process for that?
I would say for the germline variant, so we do have an annual process or revisit all the VUS and kind of issue if there's any status changes. For somatic, we don't have this process. But the reason is that somatic variants that if it's actionable, you know, for clinical use, this will already be past the time. If we rerun the analysis, it is going to take a lot of time, but the data is on the research platform, so they could still be used for research purposes. And if the, you know, if the patient relapsed, their samples would also still be reanalyzed, and we perhaps can gain more insight during that time for some of the events that we missed in, you know, in the testing of the diagnosis phase. So that's our process.
While Noemi's getting the mic, Jinghui, a very brief comment you made on one slide was pushing the results back into the electronic medical record. Can you give us some insight into the hurdles you had to overcome to be able to do that?
Yeah, so our current process is very tedious. So it's like a copy and paste by a pathologist to put the results back to medical record. So right now, currently we are changing our system from Cerner to Epic now. So Epic does have a module that allow transfer of genomic findings to Epic system directly. So we are trying to, you know, working towards, you know, utilize the API to make this an automated process for changing the, you know, direct deposit from our database to Epic at present.
Okay, thank you. Maybe last question from Noemi.
It's actually two questions, if that's okay. Thanks for this amazing presentation. Certainly really inspiring us, your work from the beginning. I have two questions, if Max allows. One of them is from the molecular Germline pipeline, and second one is related to the clinical translational component of the Germline results. So from the molecular Germline pipeline, I will be interested in knowing which type of virtual gene panel do you apply to the patients or the participant? In particular, the question is related to if you try to or adjust the list of genes based on the tumor type of the participant, or it's a tumor agnostic list of genes. This is in relation to the comment that you made around the ETV6 and the incomplete penetrance, and that's maybe why with the phenotype, this variant was initially missed. The second question is about the clinical translational component. You mentioned that the Germline reports the results were usually delivered by genetic counselors, and I would like maybe to ask you if you could briefly expand in that part of your pipeline and the return of the results to the participants. Thank you.
Okay, I'm having a little bit hard time to hear the question, so I hope that I can answer it correctly. So the first question is about the PCOM pipeline.
Can I paraphrase, Jinghui? I think it was, do you have a custom virtual panel? Do you have a custom virtual panel depending on the tumor type, or is there a general panel you apply to every patient?
Okay, so we have a general panel for all the patients, but when we decide disease relevance, there is a panel, a sub-panel for specific tumors. So obviously there are, in the germline space that we know the adult cancer related mutations may not be relevant. So this is what we have been using, we call this pathogenic, but may not be establishing disease relevance in that specific context. So the germline does have a panel, kind of virtual panel for testing. Somatic is the same way, but was mostly, the panel concept was mostly related to SME indels, but not to gene fusion and copy number changes. Because like that MAP3K case, it's not in any of the candidate genes that we studied. So in that context, we basically have to rely on literature search for fusion events to determine their relevance.
Thank you. The second question, paraphrased,
the second question is about, Mark, can you help with?
I'll paraphrase. I think the question's basis was, can you describe your experience returning those germline results through the genetic counselors or clinical geneticists, cancer geneticists? Is that right?
Yes.
Yep.
Thank you.
Thanks.
Yeah, so, this is the genetic counselors that only those consented, not every patient's family wants to know the results. So it's actually a very interesting question. So we will look at the consenting process. And we do find that African American have a reduced consenting rate compared with other ethnicity groups. So we need to do better education appeal of receiving the germline variant results from the clinic. So that's kind of the experience we can see, I can share with the group.
All right, thank you so much for giving us the insights really in how you can do good clinical science, cutting edge clinical science and discovery of that data. Right? That's what I took away from your excellent talk. So please join me in thanking Jinghui again.